Okay, let's focus a little closer on how enzyme active sites actually do function. Okay, I touched on the four things 2 lectures ago, but let's look at them a little more closely. When I say they align the substrates, okay, for a bond to be made between two substrates, the atoms within the molecules that are going to be lost, if it's a dehydration reaction, the hydroxyl and the hydrogen on a hydroxyl from two neighboring substrates have to be located close to each other so that when the bonds are strained and the hydroxyl and the hydrogen are pulled off, bonds can be reformed. Okay? If they are pointing in different directions, they are not pointed at each other, they can't be stressed, they can't be reformed. The optimal environment, as I talked about in the last lecture, it's going to be due to the R groups of the amino acids that make up the active site. Remember, some amino acids are hydrophilic, hydrophobic, acidic, basic. Well, you get enough of those R groups together that are all acidic, active site is going to be acidic. You get enough of them together that are hydrophobic, well, it's going to be a hydrophobic environment. And the R groups themselves are going to have charges, going to allow them to interact directly with the substrate so that as the enzyme changes its shape due to the induced fit, it's going to, number 3, contort, stress, twist bonds that need to be broken or bonds that need to be remade. That also ties into number 4. Now, you have to control things. You cannot have enzymes just going all the time willy-nilly. You have to be able to turn them on, turn them off. So, we've evolved mechanisms to regulate enzyme activity. Once the enzyme's made, as long as there's substrate there, it'll keep going and going and going and going. It's the Energizer Bunny. But sometimes you want to turn it off or turn it down a bit. Okay, active, yes, but not quite as active. Slow it down. So, 3 mechanisms that we see that are involved in regulation. Notice none of them are temperature. Okay, please do not mistake the last lecture saying you can use temperature. You cannot modify the temperature of your cells. First, pH. You can regulate the activity or the ability of an enzyme to function by changing its shape. Remember, pH, all those hydrogens, the lower the pH, the more hydrogens are free floating, will unwind tertiary structure. Loss of shape, loss of function. Put the pH back up near neutral again, enzyme will reform, or protein will reform, enzyme will get its activity back. Second method, come up with some molecule that's going to be able to bind directly to the body, the structure of the enzyme. By binding, it's going to change its shape. Make it work, make it not work. Or it's going to bind into the active site, but it's not a substrate. It's going to inhibit, it's going to slow things down. Or make an enzyme have to have a little helper. That's what number 3 is. The coenzymes or cofactors. These are things that are going to have to be bound to the enzyme, interacting with it, to give it its true shape so it can have a function. What scientists call coenzymes, cofactors, you've been told as a small child, these are the vitamins that you have to have in your diet. So, let's talk the second one, have something that will bind to. We call these competitive, some of these we refer to as competitive inhibitors. Competitive means it competes for the active site. Keep in mind, you have the active site. Boom. Okay, here's the active site. The rest of this is the enzyme. Substrate bounces in, bounces out, bounces in, bounces out. If it's the correct shape, it's going to bounce in, induced fit, cut, paste, twist, tweak, whatever, product comes out. But what we see with a competitive inhibitor, it kind of has the right shape, but not really, of the true substrate. So it bounces in, bounces out, bounces in, bounces out, bounces in, bounces out. When it's in, the true substrate cannot get into the active site. When it's out, now it's competing. What's going to get back into the active site first? The inhibitor or the true substrate? That's what competitive inhibitors are all about. They're competing for access to the active site. What we see is if there's no competitive inhibitor, enzyme activity is high. As you start to raise the level of a competitive inhibitor, the activity rate starts to drop, and it drops. More inhibitor, less activity, because it means there's more competition. More inhibitor means more competition, which means activity goes down, and you get this inverse. The activity of the enzyme never truly shuts off, okay? It just gets turned down. Think of those dimmer switches you have at home, you know, the ones where you turn it and you can make the lights really dim, but they're not off, or you can turn it all the way up. That's the kind of control competitive inhibitors are all about. The higher the concentration of inhibitor, the more control, the less activity you're going to see. Non-competitive inhibitors are going to bind somewhere else to the enzyme. They're going to bind somewhere else, and they're going to change its shape. Loss of shape, loss of function. So as I said, with competitive inhibitors, it's never truly turned off. It's brought down its activity levels. The more inhibitor, the lower the activity, but it's never truly off.